week, Surfing the MASH Tsunami brings you three major stories from the world of steatotic liver disease. This weekend, we're bringing you each of these stories separately so you can listen over a shorter, discrete period of time. Hope you enjoy them.
For the last 5 years, MASLD stakeholders, whether manufacturers, trial sites, providers, payers, patients, or investors, have turned to Surfing the MASH Tsunami to keep track of drug and device development, epidemiology, public health initiatives, and practice patterns. This weekend, we are sharing the three main sections from our episode 7. Roger will provide more information after these notes from our sponsors.
Surfing the MASH Tsunami is sponsored by these healthcare-related organizations: Liver Right and the Liver Right Scanning Network. On April 15th, 2025, Liver Right will open America's first virtual specialty clinic for liver disease, offering adults in all 50 states video appointments and helping GI practices and health systems triage and treat the swelling numbers of patients seeking MASLD and MASH testing. Liver Right offers same-week video visits and accepts insurance. For more information, contact Liver Right, that's liver, and then RIGHT at www.liverright.com.
Tawazin Health, your partner in global liver health innovation. At Tawazin Health, we specialize in providing small or large-scale access to FibroScan examinations for liver health-focused early detection, clinical trials, independent research, and collaborative projects with clinicians and FibroScan providers increasingly throughout the world. As industry-leading experts, we design and deliver high-quality FibroScan services because we know that accurate, high-quality, reliable data drives better outcomes. To learn about our sister lifestyle app, mylife365.me, click the Tawazin Health link on the Surfing MASH homepage. This week, our roundtable on drug development includes Roger, Jörn, Louise, and hepatologists and key opinion leaders Naim, Alkhouri, and Sven Francque.
This week, our special section from the Roundtable on Drug Development includes Sven Francque, Jörn Schattenberg, Louise, Campbell, and me, focusing not on specific drug development issues, but on how to think about MASLD as a disease and the role that NITs can play in treatment and clinical trials. Louise starts this segment by asserting that our goal is not simply to reduce liver fat, but to take actions that help the patient become more metabolically healthy, which may include trials and counseling more than simply drugs, and may focus on the overall patient rather than simple liver fat. Sven concurs. After a brief detour for me to discuss the potential value of glucagon, Jörn shifts the broader conversation to focus on the role of NITs, particularly transient elastography, in patient management and drug testing. The entire group lists some benefits and future uses of NITs, ranging from being sufficient by themselves for a drug to be granted conditional approval, to the idea that repeat measures can be an important part of assessing and driving success in therapy.
So when does somebody become metabolically healthy and liver healthy as opposed to driving just weight loss? Because once we start to set targets, people get frustrated if they don't reach them. We set failure into the mechanism, which is not what we're aiming to do. And a lot of people want to keep their curves. They just want to be metabolically more healthy. There is that fine balance without driving and supporting people's change in health status and knowing more. We've got more tools at our disposal. Can we use them better to find those levels? Traditionally, I will see a lot of people with type 2 diabetes with steatosis grade 2 by FibroScan, for example, with very little fibrosis or stiffness in their liver as evidence. So where do they become metabolically unhealthy as opposed to metabolic drivers of disease? So I think non-invasive technologies now offer us all of these sort of facilities, and the trials are gathering that data. And there are some very exciting medications in earlier trials coming up that have lots of different mechanisms. It's an exciting area to be in.
Yeah, absolutely. And I think, Louise, what you said is so important. It's the metabolic health that is in the center of what we want to achieve.
That sounds right. And to go past that, remembering that my last science course was high school biology, but that I do know a fair amount about statistics. The one thing you can predict about any straight line is that it will eventually turn into a curve, and that there will be at some point a diminishing effect of weight loss. That's just got to happen. Where that curve bends, that's not clear yet, but I think it's absolutely clear that one of the things that you get by bringing glucagon to the party is that you're now working on at least two different ways of improving liver health. One, weight loss, which will curve at some point, and the other, the direct liver effects of glucagon, which make a lot of sense to be more powerful or to potentiate greater change faster than a simple linear move on weight loss would.
Yeah, but it's very important that we're now starting to get the data where we have indeed the comparison and demonstrating that it indeed has an added value, because as long as you do not have the data that really allow for a straightforward comparison, it always ends up in discussions about numbers and small differences. But now we are getting data about true differences that are very important.
We've lost Naim, though. I mean, the other thing, and we gave him a little bit of airtime because he had to drop off to clinics, is he mentioned briefly, and I think this is a game changer, we've talked about for the longest time on this podcast, going back to season 1, is running a trial with non-invasive tests, enrolling patients based on NITs, following them to outcome. And we haven't quite seen that yet, but I have also the feeling that he shared that with the situation or the time we've progressed so much in understanding NITs and defining the populations that are at higher risk to develop outcomes. We've seen more outcome data linked to NITs, and by doing that, we can actually calculate the number we need in a trial and the time we need to follow them up to actually see those events. And that would be, of course, a game changer, having a non-invasive trial run to full outcomes, trying to modulate the course of the disease using a highly potent drug.
Yes, it is exciting that is coming. And to be fair, it's how hepatology has managed its care for a long time. We don't biopsy unless we need to. We try to be as less invasive as we can in this area. Do you think the trials are now starting to be more reflective of the way hepatology has gone over the last few years?
See, the field has pushed for that for the longest time. There's the recent Liver Forum meeting in Washington was all about that. We learned a lot about NITs and the qualification of NITs as surrogates, even for surrogate approval. And I think this is a little bit further down the line. But following the patient to outcomes, it's something the FDA has been pushing companies, as far as I understand, or is at least suggesting to the field for the longest time. They want to see those outcomes trials and would be much more comfortable compared to conditional approval, of course, than for a drug. And I think we're very closer to that. Even NITs as a surrogate, well, that needs more discussions now still.
Yeah, I think we still have a way to go, but I think we have indeed made quite some progress there. It's too early, I think, to really align clinical trial with routine clinical practice, but because, before something can enter clinical practice, you need solid proof of efficacy, and it's not always that easy, so it's probably a little bit early indeed, besides the larger NIT-based trials for a long time for outcomes, but still, I think we're making progress for sure.
I'm thinking of the clinical trial data from the VCTE study group, work Vincent Wong has pioneered, pulling a lot of people together, and Sven, you might have been part or you contributed in parts to that, looking at the greater than 10 kilopascal cutoff population and the number of events we're seeing there. Louise, you have a lot of experience with FibroScan. Of course, in clinical trials, you might want to combine the FibroScan, and that's a whole different discussion, with a second NIT to reduce the noise a little bit, because it has some variabilities. However, still the testing and the experience of the operators has improved so much over time that, in clinical practice, we're basing decisions on changes, maybe not 1 kilopascal, but a number, in a ballpark. And the data that's been brought forward is the change of 30% that really does change the disease trajectory in our patients. I think even below that, in clinical practice, I would consider an improvement for my patient at the individual level. Of course, it reinforces the patient to actually keep up his behavioral change or whatever have led him to achieve that reduction in VCTE.
I think that's right, but I think there's a bit of caution that I'm going to put in here, is that the more that we drive this, the more that we think that FibroScan is an easy tool. The more that we do one, we see one, do one, teach one. That is not what VCTE is. It is not accurate. I saw a recent publication of a big article in the Journal of Hepatology where they were very clear. They stated that the people trained on FibroScan had been trained on the one-day EchoSense training and had at least performed 5 FibroScans. That does not quantify an accurate clinical study. They might have got a fairly good outcome, but there's a very good hepatitis B study from China where they looked at over 1,000 FibroScans, and in every state of that, they detailed what their IQR was. They knew that over 85% of their patients had an IQR of less than 0.15, so less than 15%. Therefore, it correlated really, really well with the liver biopsies. So we need to be careful when we talk about discordance in FibroScans scans that somebody can recognize an overreading trace. Somebody can recognize an A wave. Somebody can recognize a Y wave. You are not going to recognize that in only a day's training and 5 scans. We have governance documents where we supervise with expert supervisors for at least 30, and they talk to us about them. They look at those traces. We can be more accurate if we control the accuracy of VCTE going forward. That can correlate better with what our outcomes are. And most of our biopsy studies, we will know from the IQR, which is never published in a trial, of which IQRs correlated better with the liver biopsies. It is going to be the ones on statistic tissue. Our study at Imperial, when we did it, we did ultrasound, we did a shear wave, and we did biopsy with FibroScan all at the same time through the same point. So it correlated very well, and we were able to plus and minus people's weights to be even more accurate. So I think my air of caution is the increased need to things like VCTE is going to drive a watering down of the qualifications and the skill of the user and the supervision level. Now that could come back and undermine some of the beautiful work that's being done in these trials if we don't really nail down what do IQRs mean to us and how accurate we want. And we know we've got technology now that's 42 times more accurate than some of the devices still being used in trials in some locations around the world. So there's a great deal of need to say, what do we want to do as a profession and a speciality about controlling the non-invasive technologies and their accuracy levels where we can.
I think, Louise, that's a very, very important point, because as you know, there's also a push now to go more to point of care technologies, not just VCTE, but also the other elastography techniques, positioning it even in first-line practices, which is, of course, in a way good for screening case finding, but it increases even further the concern about the qualification of the people using it. And I think we cannot emphasize enough how important repeated measurements are also, because it's not just a 2-point measurement that will tell you whether it's going better or whether it's worsening. I think you need sequential measurements, and then you can see an evolution in the good or in the bad direction that is more trustworthy than when you just compare two different time points.
Yeah, I think that's absolutely a fantastic point, and I think that's why we take skilled users into primary care, because they don't know the device, they don't want to hold it, and they don't have the skill to have those conversations. So we take it in to where you need it. But the other thing that you said, there is this sequential scanning. What we're now seeing in clinical locations of primary care is that brief intervention and the experience of a FibroScan in a primary care will move somebody's KPA from around about 11 to 9 or from 10 to 7, because you have the brief intervention, you then have that patient make a change. By the time they get to referral to a specialist, because we've hit that cutoff of 8, they've changed significantly because they've actually responded to that scan in the location. So I would suggest, if we're looking for referring, do exactly what you've suggested. You do serial scans in primary care. 80% of them will improve, probably the 80-20 rule, if it applies. They're going to improve. They don't need specialist care this time. Take the 20% who don't improve over 3 to 6 months and move them into specialist care, because otherwise we are going to send a lot of people with 8 KPA and above to specialists like yourself, who again will come back into primary care, who can learn to manage it better.
The EASL guideline on the recommendations of MASLD actually allows for that with the intermediate FIB4 category of either direct referral or managing according to your system. And I think it's important to actually manage them the best we can, because we can only provide liver-centric care to so many people to not clog up our referral pathways. And we want to see the right patients, of course. So I agree with Louise and Sven that it's our responsibility to provide that standard of care. In clinical practice, it's not as easy because not everybody has that access to nutrition. Physiotherapy was one of your questions earlier, Louise. I think in the clinical trial setting, this is much more advanced and we can offer more, but it's beyond the reality of many or most patients to actually have a good nutritional therapist access and consult them. And that's, of course, the downside of this, and that's why the progress in drug development is so important in this round table, more than important.
On behalf of the surfers, our guests and our sponsors, Madrigal Pharmaceuticals, Liver Right, and Tawas in Health, thanks for joining us this week.
Please join us again next week when we conclude our roundtable on the Emerging Trends Conference, while our newsmakers and experts explore current issues and major ideas.
And until next week, stay safe, surf on, and we'll see you on podcast. Bye-bye now.